Thanks a lot, Josh. Being from Northern California, I'm supposed to say derogatory things about Southern California, but I actually really love it down here. I think it's great, except for your basketball teams. So I'm going to start with a bit of a historical approach, and because I think that'll help get us organized. And then I'm mostly going to talk about where I think things are headed. So if you haven't figured it out, I have virtually nothing to say to you that's going to be useful today. I think if you're caring for patients with Alzheimer's disease, I guess there are a lot of things you should know about imaging, but most of them aren't going to be terribly helpful. So that's my caveat in advance. So I think, the earliest kinds of imaging that we could get on people were structural brain scans, first CT scans, then MRI scans. And then beginning in the 1980s, we began to do glucose metabolism imaging with FDG, or fluorodeoxyglucose PET. From the 1990s on, we started with functional imaging using MRI, functional MRI, and so forth. And then from about the 2000s, we really started what I would say is the modern era of molecular imaging, looking at amyloid and tau in PET scans and looking at these protein aggregates. And all of these techniques, interestingly, are still really important in research evaluations of dementia patients. So that's why most of what I'm going to have to talk to you about today is how these are used in research, and I think how they're driving the field towards therapeutics. So you heard about this clinical diagnosis. Actually, it was great to see this. This is when I started my career. And when I look back on this, I'm kind of amazed that I started my career in Alzheimer's disease at a time when we didn't really even agree about what Alzheimer's disease was. And these were clinical criteria. They used the term probable Alzheimer's disease, and they were very, very important. But things really have shifted. And so you've also heard about this, this new approach, which was published a year ago, of this new research framework that I'll get to in a moment, which has almost entirely been driven by biomarkers and brain scans. And this conceptualizes Alzheimer's disease as a biological disorder rather than a clinical syndrome. And so this took us 34 years. So I like to point that out when we look at, how much progress we've made. We really, in a way, we're just getting started with the modern era of Alzheimer's disease research. So you've heard about the neuropathology. I don't really have to go into this. It's great that you're already familiar with this. Probably many of you knew this. I just want to say that the amyloid and plaque pathology, or the amyloid plaque and tau pathology, the protein aggregates, but there's also this other thing that you've heard about a lot called neurodegeneration. We usually look at this as brain atrophy. It can also be changes in glucose metabolism or brain function. And again, as you've heard, this seems to be sort of the end result of all the pathological processes that are playing out in the brain, and it's the process that bears the closest relationship to cognitive function. So when you see these shrunken brains and severe changes in structure and glucose metabolism, individuals with these kinds of pictures are frequently suffering from cognitive impairment or dementia. So I just want to say one thing about PET, to put PET in perspective. It's actually really a complicated kind of thing. You need to have a cyclotron to make the radioactivity that goes into a PET radio tracer. These are usually carbon-11 and fluorine-18 that have pretty short half-lives. And then you have to make the molecule. You have to stick those radioactive atoms onto a molecule, so you have to do that fairly quickly. Carbon-11, by the way, has a half-life of 20 minutes, so you really have to work fast. And then these are injected into a person. They circulate, go through the circulation. The individual's in a PET scanner, and we can make the image. So one of the nice things about this, though, is that because fluorine-18 has a sort of a longer half-life, you could have a central radiopharmacy that makes radioisotopes makes radio tracers labeled with fluorine-18, and you can ship those around in an area. So, for example, cancer diagnosis now is routinely done with fluorine-18 labeled radiopharmaceuticals in hospitals. So theoretically, there are very, very few barriers to getting this technology out into the community. The only real barrier is whether or not it's useful. So I didn't mean that as a joke, but I guess it is kind of sad. Okay, so these were some of the first studies that we did, and really some of the first sort of beginnings of PET imaging were with fluorodeoxyglucose to look at glucose metabolism. And what you see are these reductions of glucose metabolism in the areas that these arrows are pointing to in this patient with Alzheimer's disease. And we think this largely reflects activity at the synapse, the areas where the neurons talk to one another. And so we think this is reflecting the function of the brain having to do with normal neuronal function. And this is a biomarker, and it's a biomarker of neurodegeneration that reflects synaptic dysfunction. Is it diagnostic? Well, sort of. This is an example of a glucose PET scan in Alzheimer's disease, the one I just showed you basically, and a patient with frontotemporal dementia, the FTLD on the top. And you can see the frontotemporal dementia subject. Are these showing up? Yeah, good. I was really feeling sorry for the people on that side of the room. But you can see the frontal lobes in this frontotemporal dementia patient showing lack of glucose metabolism, whereas in Alzheimer's disease, this is in the parietal and temporal lobes in the back of the brain. And so this was actually one of the first brain scans, the FDG PET scan, that was applied to diagnostic use in Alzheimer's disease, and it's still been used somewhat. And in fact, Medicare will reimburse for glucose metabolism studies if you're trying to differentiate Alzheimer's and frontotemporal dementia. It's probably the only area that it's useful. And again, you have to argue how useful is it, since we don't really have treatments for either of these. But I want to show you the sort of another view of this, and this is a map of the changes in glucose metabolism, sort of laid out on the brain, because the pattern of these abnormalities is really interesting. And it was interesting to me when I started my career, 30-something years ago, and it's still very interesting. And what's really interesting about it is that Alzheimer's disease glucose metabolism affects these parietal and temporal lobe regions, and that's where the metabolism goes down. And if you look at the MRI scan, it's very, very similar. The brain shrinks in very much the same regions, although the one slight difference is that you see a very big change in the volume of the hippocampus and the related structures in this part of the brain, which we call the medial temporal lobe. And this is the part of the brain where memories are formed. This is very important for making memories. And so these changes in metabolism and cortical thickness have been known about for a long time, and we really haven't understood very much about how they come about. What we do know is they tend to be related to the clinical state of the patient. So if these are more severe, patients generally have more severe symptoms. And there is a very similar pattern to this sort of what we call regional vulnerability for metabolism and atrophy, and I'm going to come back to that as we go through the talk. So things really took a big jump in the early 2000s, when the first specific amyloid imaging agent was developed by two scientists, Chet Mathis and Bill Klunk at the University of Pittsburgh. We owe them a great debt of gratitude. They figured out that you could take this compound that was used in pathology studies, like Dr. Schneider just talked about, and you could modify the compound a little bit so that it got into the brain and label it with this carbon-11, inject it into a person, and then see the amyloid in the brain, these fibrils that form the plaques. And so this really broke open a whole new approach to diagnosing Alzheimer's, or at least being able to visualize amyloid. And though this was done in the early 2000s, what followed were a number of other compounds that were labeled not with carbon-11, which is really, really difficult to use, but with fluorine-18, which again now can be made and transported. And so there are now 3 of these compounds, flutometamol, florbetadine, and florbetapir, that are actually approved by the Food and Drug Administration. This 4th one is still sort of in a developmental phase, or it hasn't been approved. But these 3 are approved by the FDA, and as a number of people have said, you can get a PET scan, a doctor can order them. They're generally not ordered because Medicare won't pay for them, and the reason for that is because they don't really have, or at least Medicare, the Center for Medicare Services has decided they don't have a big enough impact on patient care and outcomes. But I think notwithstanding that, the fact that they can be manufactured and delivered pretty much anywhere in the United States has had a huge impact on the way we think of doing clinical trials, and I'll get to that toward the end of the talk. So I just want to contrast, what I've taken you through. Since the 1980s, we had glucose metabolism scans, which showed these characteristic locations of abnormality. And then now we have these amyloid imaging scans, which are not remarkable for like a subtle visualization of where the abnormality is. They're almost black and white. There's virtually no binding of this radioactive compound here. These green and yellow colors are kind of a background, and these hot red colors are the amyloid. I didn't do anything tricky to this. This is what the scans can look like, and so we're really seeing a phenomenally different kind of signal when we look for amyloid than when we look for neurodegeneration. And I just want to sort of hammer this home here that to make these points. So these neurodegeneration markers, whether they be changes in brain volume or atrophy or changes in glucose metabolism, are telling us something about the fact that the brain has been damaged. But they're non-specific. They don't tell us why the brain has been damaged. The shrinkage of the hippocampus, yes, it's associated with Alzheimer's disease, but as Dr. Schneider told you, it's also seen in TDP-43, frontotemporal dementia, vascular disease, and so forth. They may be very complex to interpret. It's a lot easier to see that black and white amyloid jumping out at you than seeing a subtle pattern. The neurodegeneration, though, since it tells us where the brain is damaged, it tends to correlate very well with symptoms, where the molecular biomarkers don't necessarily do that. And although these are used in drug development, I think there's a lot of argument about how useful they are. So the molecular therapies are quite different. As I said, they're not tracking damage, they're tracking the specific pathology. They're not non-specific, they're specific. They're telling us about these protein aggregates. Interpretation, it's not trivial, but I think it's more straightforward. It may or may not correlate with symptoms, and not only should they be useful for therapeutic testing, but I think they are, although I guess the jury's still out. So let me turn to tau. There are a bunch of different tau ligands. This slide is actually a year old, and it's not even up to date. There are a number of different tau radiotracers, just like there are a number of different amyloid radiotracers. I should say that in the imaging field, we've figured out how to make the amyloid imaging images, regardless of what radioactive compound is used. We've figured out a way to make them comparable to one another. We haven't been able to do that yet with the tau tracers, but hopefully that's coming. So most of I think all of the images I'm going to show you are taken with this one compound that's called fluortaucipir. It's the tau imaging agent that's been available the longest. And it's a kind of a different picture than the amyloid PET scan. So let me show you these. Excuse me. So this was one of the first studies we did with this tracer in my lab. And we have 4 groups of subjects, and the idea is we're going from what we would guess to be the least amount of tau to the most amount of tau. So the least amount of tau is young people. So in my lab, we study aging. We study Alzheimer's. We also study aging, and we study people in their 60s, 70s, 80s, and 90s. And there's no control. You can't say, I want a 60-year-old control subject, because we don't know what's happening in the brains of 60-year-olds. So our true control subjects are college students. And coming from Berkeley, So we only had 5 at this point, but I just want to show you. This blue colors are the background. There's virtually no binding of this tau PET ligand in these people who are ranging in age from 20 to 30. And so that's a truly negative tau PET scan. So these are older adults. We say they're PIB negative. PIB is the Pittsburgh compound B. It's an amyloid imaging agent. So these people have no amyloid in the brain. But you can see just by comparing these 2 images that there's some tau in these people's brain. There's a little bit of tau here in the temporal lobe. A little hard to see, but it's there. These are our older adults who have amyloid in the brain. Now you've heard that this is something that is not that rare. These are older people. They're cognitively normal. They're completely normal. They have amyloid in the brain, and they have a little more tau. And you can see that if you compare the amount of tau here in the temporal lobes, it's gone up certainly compared to the 20-year-olds, but also compared to the people who don't have amyloid in the brain. And then here are patients with Alzheimer's disease. So they have a great deal of tau in the brain. And And if you sort of map this out and ask, well, where is the tau in general when it's elevated, it's sort of in this zone here. And I think you can remember back to those scans I showed you of the glucose metabolism and the cortical thinning, and it tends to be fairly well paralleled with where the brain is shrinking and where glucose metabolism is going down. So I just want to say one more thing about Alzheimer pathology and normals. I'm going to keep coming back to that, but you can certainly have a lot of amyloid in the brain and be cognitively normal. But I don't think, at least at this point with all of our tau imaging, that you can have this much tau in your brain and be cognitively normal. And that's one of the big differences between amyloid and tau, is that extensive, widespread deposits of tau are generally not compatible with normal cognition, whereas they're quite compatible with normal cognition if it's amyloid. So is this useful for diagnosis? Well, there isn't a lot of data yet, and there especially is not a lot of data with autopsies. We heard from Dr. Schneider that that's what we need to confirm a diagnosis. So I don't want to put too much weight on these studies, but this was a study that combined clinical diagnoses in over 700 people from a number of centers. And what you can see is that there is a lot of elevated tau in patients with Alzheimer's disease and mild cognitive impairment, and it had pretty high sensitivity and specificity in telling these Alzheimer's types of dementias and cognitive decline from other disorders. So again, I think until we have large autopsy series, the actual clinical utility is going to be unclear, but at least these very large, high amounts of tau are generally not compatible with being normal, and they don't seem to be compatible with a multitude of other dementing illnesses. So just to put all this on one slide before I sort of move on to some of the more research applications, I just want to show you what these look like so you can see them all together. So this is a single individual with Alzheimer's disease, and this person has a lot of amyloid, and it's in the parietal lobe, temporal lobe, frontal lobe. It's really pretty much everywhere. There's an area here that's spared if you're really interested in paying attention. This is the sensory and motor area of the brain, and that's an area of the brain that's generally spared in Alzheimer's by all these processes and doesn't show clinical involvement either. So again, I want to stress, yes, this is a person with Alzheimer's disease, but it wouldn't be unusual for a cognitively normal person to have that much amyloid in the brain. This is the tau ligand, and it's certainly more restricted than the amyloid ligand, but there's quite a lot of it in this person's brain, especially compared to some of those, the slides I showed you previously, where there is, so this individual has lots of tau in the parietal lobe and the temporal lobe, the medial parietal lobe, and again, this kind of tau is very likely, this amount of tau is very likely to be associated with cognitive impairment. Here's the glucose scan showing areas where glucose metabolism has declined, consistent with synaptic dysfunction, and here is the cortical atrophy. And again, I'm going to come back to this later. These three scans, the tau, the glucose metabolism, and the MRI atrophy, have a lot of commonalities in terms of what the patterns are. And so I'm going to, as I said, come back to that. So I want to tell you about a study that I think was a landmark that was published in the New England Journal a number of years ago. This was done by the group at Washington University led by Randy Bateman, and it's a study in a group of individuals that have been called the Dominantly Inherited Alzheimer's Network, or DIAN study. And this was really a fascinating study. What they did in this study is they collected people who have these gene mutations that you heard about from Dr. LaFurla at the beginning, individuals with these autosomal dominant mutations, and they gathered them together in multiple centers around the world, and they collected biomarker data. They measured amyloid, tau, and so forth. Now, one thing actually I should tell you is that I'm talking entirely about amyloid PET scans and tau PET scans, but you can measure amyloid and tau in the cerebrospinal fluid. I'm not an expert on that, and we have some data, but I'm really not going to talk about it. But most of the things I'm telling you about with PET are transferable to spinal fluid measurements. And so some of these measurements that they did in DIAN on biomarkers were done with PET scans, others were done with spinal fluid measures. So they collected symptomatic and asymptomatic people. So in other words, they examined people with biomarkers who carried the gene mutations but didn't yet express symptoms. So one of the things that's interesting about these families is the age of onset is preserved across generations. So if you come from one of these autosomal dominant families and your parent got sick with Alzheimer's disease, say at 50, the chances are that you'll get sick around 50. So if you have a PET scan for amyloid at the age of 40, we can say you're 10 years before the age of onset if you have the gene mutation, right? So what they did is they calculated the changes in all of these biomarkers in relationship to the age of onset. And that's what this line here, 0, at 0 means that's the estimated age of onset for all these people in the study. And what these results actually show is that the first thing that changes is the amyloid starts to go up in the PET scan. And actually, it starts to go down in the spinal fluid about the same time. The positive spinal fluid is a low value, we think, because it's being absorbed into the brain, not floating around in the spinal fluid. So I just want to stress this, that the very first thing that goes up is amyloid in the brain, and that happens around 20 years before the expected age of onset of Alzheimer's disease. And the next thing that starts to happen in this study was the tau started to go up in the CSF, and pretty close in time, glucose metabolism went down, and the brain shrank, and the very last thing to change is this black line, which is the cognitive tests. So the cognitive tests don't change until just a few years before the onset of the disease, but the amyloid changes 20 years before. And so this has been very current and important information motivating this idea of this amyloid hypothesis or the amyloid cascade hypothesis. This certainly isn't the start of the amyloid hypothesis by any means, but it's an interesting test of it in humans, and I think biomarkers really enabled this. So this evidence, along with a lot of other evidence, a bit of which I'll show you over the next few minutes, has really pushed this idea that Alzheimer's disease can be thought of as a biological disorder and not simply a clinical syndrome. And that's led to this research framework. And I'm glad that Dr. Sulzer mentioned this already. There are 3 pathological processes. I've been talking about all of them. Amyloid, which is the A, tau deposition, which is the T, and neurodegeneration, which is the N. And we can classify people by whether they have this. And so I just want to point out that if an individual is amyloid positive, we say that they're on the Alzheimer's continuum. That means that they have something related to Alzheimer's disease. But if they're positive for amyloid and tau, then we say they have Alzheimer's disease. Now, that's a very controversial notion. I was one of the co-authors of this paper, so I guess I have to defend it, and I will. I think this enables a lot of things that we couldn't do before. We can study people who are amyloid and tau positive, for example, with amyloid or tau-lowering drugs, in the hope that we might be able to intervene because we think they will get clinical symptoms of Alzheimer's disease. And I guess to point out, this is really meant for research. And I realize that telling people they can't use it isn't something you can do, but it's not meant for clinical care. It's not meant to be a way to diagnose Alzheimer's disease in the clinic. I don't think someone with a positive amyloid scan and a positive tau scan really should be told that they have clinical Alzheimer's disease at this point, because they don't. So is amyloid and tau useful? And what is it telling us about both the patients and the biology? So one thing it certainly tells us is that amyloid status predicts clinical conversion. So these are normal people who are being followed to mild cognitive impairment. These are mild cognitive impairment patients who are being followed till they get Alzheimer's disease. And you can see as this line goes down, it means more and more people develop MCI. As this line goes down, it means more and more people develop Alzheimer's disease. And you can see if you're amyloid positive, you have a higher risk of getting Alzheimer's disease from mild cognitive impairment and a higher risk of getting mild cognitive impairment if you start out as normal. So that's pretty good evidence. I'll spend a minute on pointing out that really where the action is in normal cognitive aging. And that's because we think one of the reasons that maybe clinical trials have failed is because lowering amyloid in someone who has full-blown Alzheimer's disease is not likely to be effective. And so we have to study the very beginnings of Alzheimer's disease. And while it's great to study this in people who carry gene mutations, that's a very, very small proportion of the people at risk for Alzheimer's disease. So the way to study it, many of us believe, is to study normal, healthy, older people. And so these plaques and tangles are common in healthy older people. This is just a graph from a pathology study showing that by the time people get into their 70s, this purple line is the neurofibrillary tangles. By the time people get into their 70s and 80s, virtually everyone has these tangles in their brain. But here's the thing, they don't have them everywhere. They tend to begin, we think they begin, of course we don't know because most of these data are from autopsy studies. We think they begin in this red area, which I've already talked about, and that's the medial temporal lobe memory system, or the hippocampus. And of course, we can see that on MRI scans and on these PET scans. So we can see the tau there. And so we can begin to look at the very beginnings of Alzheimer's disease, or at least the beginnings of tau deposition, with PET scans and look at this medial temporal lobe brain region. And we can see the amyloid throughout the brain, as I've been telling you. So how well does amyloid predict cognitive decline in normal aging? I'm going to rush through this slide because I just want you to see the main point, which is a very nice study that was done a couple of years ago in this project that I'm part of called the Alzheimer's Disease Neuroimaging Initiative, or ADNI. And there's now several hundred subjects in this study who've been followed for an average of about 5 or 6 years. And these are all people who started out normal, and what you're seeing here is a cognitive test score in individuals who have normal amyloid and elevated amyloid, or amyloid negative and amyloid positive. And you can see these lines are beginning to diverge. And this is another cognitive test score where a higher score is worse. So these people are also doing worse. So the amyloid positives are doing worse than the amyloid negatives on both of these tests. But what I want to point out is these lines here don't really start to diverge until the people have been followed for about 4 years. So again, these are older people. It depends on how old you are. It depends on how long you're followed. But I think there's emerging evidence that if you follow people long enough and if they're old enough, amyloid is a predictor that something bad is going to happen. So one of the reasons that I think, and I think many people think, these processes take so long is they're not direct. So it's not that amyloid has a direct effect on cognition. It has an effect, but it's weak. It takes a long time to come out. And it probably only is strong if the individuals are older. And what happens in the intervening time is that tau is deposited, and that this tau is leading to changes in the brain in terms of degeneration, and that leads to this cognitive decline. So I'm just going to take you through a few bits of evidence for this and try to convince you that this pathway that I'm articulating here is a good model for what happens in Alzheimer's disease, and it's really a model that's built on biomarkers, so it's testable with biomarkers. So let's look at this amyloid and tau. So these are data from the Alzheimer's Disease Neuroimaging Initiative again. And we're looking at 3 brain regions. Here we're just looking at the tau in the medial temporal lobe. Here we're looking at tau in this sort of mid-sized brain region. And here we're looking at tau throughout the whole brain. And I'll just say again that if you have tau like this, it's pretty unlikely you're going to have dementia or Alzheimer's disease. It's pretty unlikely you're going to be cognitively normal. If you have tau like this, it's pretty likely that you're going to be cognitively normal. And if you have tau distributed like this, you could be either. So that's where it gets complicated. But the point I want to make is in all of these squares, we have men and women separate. We have impaired, unimpaired, that is cognitively normal, or cognitively impaired. And in all of these, there's a positive trend with amyloid. So as amyloid goes up, tau goes up. So that has been found in this ADNI study. It's been found in virtually every group that's looked at it. The more amyloid you have, the more tau you have. And what's really interesting is if you look very early at these cases who have very, very low levels of amyloid here at the very beginning, it's true there as well. So in all of these brain regions, individuals here who have very low levels of brain amyloid, the higher the amyloid, the more tau there is. So tau and amyloid are related. So what's happening with tau? Where is it depositing and where is it going? So here is a figure that shows the amount of tau in the brain that's correlated with age. And so the colors that are mapped out on this particular image are telling you tau goes up in these colored areas as people get older. And what we're seeing is the same thing that I've been telling you from the pathology studies, which is as people get older, the tau goes up in the medial temporal lobes in these memory centers of the brain. What happens as amyloid is deposited? This is where tau goes up as amyloid is deposited. It's throughout the surface of the brain in the temporal lobe and the parietal lobe. And so what is happening is that as we're getting older, we're depositing tau in these medial parts of the temporal lobe in the memory areas. But as amyloid starts to come along, it seems to either push or pull or somehow make the tau spread out of these memory areas into the rest of the brain. So how is that related to the next part of the story, neurodegeneration? Well, I'm probably going to rush through this kind of quickly, but this is an example of where glucose metabolism and tau are correlated. So what I'm showing you here is as tau goes up in these brain regions, in our normal older people, these are our normal older people who are actually positive for amyloid, as they have more tau in these brain regions, their glucose metabolism starts to go down in these brain regions. And if you remember back to the map of glucose metabolism I showed you in Alzheimer's disease, these correspond. So what's happening is the deposition of tau in normal people, if there's amyloid present, is driving down glucose metabolism. It's beginning to cause neurodegeneration in people who are cognitively normal. And we see very much the same thing with tau and brain shrinkage. So this is the map of tau change over time in a group of our cognitively normal older people. This is the map of brain shrinkage over time. This is the overlap, so you can see the brain is shrinking in pretty much the same regions where the tau is depositing. And this just shows you that they're correlated, that there's a correlation here. But what I want to show you, again, is the map of cortical thinning that I talked about at the very beginning, very much also parallels these maps of atrophy and tau. So the relationship between tau and atrophy, it fits together in the surface of the brain. I'm going to skip this slide. And I'm just going to tell you one more bit of information about this. Though there are interesting variations of Alzheimer's disease, and there's types of Alzheimer's disease where patients develop the disease at a young age, and they may present with symptoms related to aphasia. So this is called primary progressive aphasia, or visual symptoms. This is called posterior cortical atrophy. And this is the map of amyloid in these three groups of individuals. These are Alzheimer's patients who have memory disorders. These are Alzheimer's patients who have language disorders. And these are Alzheimer's patients who have visuospatial disorders. And what I'll tell you is that these all look the same. There's no relationship between where the amyloid is and what kind of symptoms the patients had. And it's sort of mapped out here in the brain. There's nothing very impressive. But here's the maps of tau. These are the memory disorder Alzheimer's, and these are the language disorder Alzheimer's. So it's a little subtle, but this is the left side of the brain, this is the right side of the brain. In the language disorder Alzheimer's patients, there's more tau in the left hemisphere than in the right hemisphere. And in the patients with visuospatial disorders, there's more tau in the visual centers of the brain than there are in other places. So the tau correlates with the patient's symptoms in Alzheimer's disease, and the amyloid doesn't. And then finally, this is the relationship between tau and memory in cognitively normal older people. So what you're seeing here is a map where we took our normal subjects. There were something like 80, all completely normal older people, and we ran a correlation between their performance on memory tests and the amount of tau in the brain. And what you see in these little spots are areas of the brain where as the tau went up, the memory went down. So these are, again, normal people, but they're depositing tau in this area that in the medial temporal lobe. This is the entorhinal cortex, very close to the hippocampus. And as people had more tau in these brain regions, their memory declined. And so this shows the correlation simply between tau and memory loss. These individuals with the higher tau had poorer memory performance. But again, these were all people who were performing at the normal level. So one way to think about this, and I think it's kind of early to, to stick a flag in the ground, is that maybe tau in the medial temporal lobe as we get older is contributing to some of the memory loss we experience as we age. I would not say this is the only thing. You've heard a lot about vascular disease. I'm sure that's going on. There are other pathological processes as well. So memory impairment as we get older does not have a unitary cause. I wouldn't want you to come away with thinking that. But I think tau may well play a role. But then what happens is that the amyloid begins to deposit. This tau, again, spreads out of this brain region into other parts of the brain. So I'm going to just sort of end with this, this message about clinical trials. So there are a lot of failed clinical trials. And if you go back and look at them, the first clinical trial that was done for an amyloid-lowering agent didn't do PET scans on people before they got in the trial. This was a drug called bapineuzumab, and you could get into the trial without having a PET scan. And it turned out they did do PET scans, though, but they just didn't use them for entry criteria. It turned out that when they looked, about 15% of the people in the study didn't have amyloid in the brain. So they were treating patients with Alzheimer's disease with an amyloid-lowering drug, but they didn't have amyloid in the brain. And of course, by our current criteria, they didn't have Alzheimer's disease. So that's a problem, but that's been fixed. So now pretty much all clinical trials, I can't imagine there are any trials of amyloid-lowering drugs that don't require an amyloid scan at the beginning, but we can also do amyloid scans at the end. And this was the study that there's been so much talk about in the last week. It was the drug called aducanumab, and this was a phase 2 trial. It was not powered to detect a clinical response, but they found one. So what they found, first of all, is the amyloid was cleared. These are ascending doses of the drug, and they found the amyloid was cleared out of the brain. This is very common now. These antibodies directed at amyloid can remove amyloid from the brain. We know that. This was the remarkable aspect of the study, is that this is a score where a lower score is better, and as the dose of the drug went up, people improved. So everyone was very excited by this, but it wasn't enough to get approval by the FDA because it was a small study. It was just designed to sort of test the waters. And then, of course, in March, the study was terminated, and now has been sort of they've decided the study was positive. And so I'm telling you this story. Actually, it's funny. I turned my slides in late, and I'm glad I did because I had a big red X over this to tell you that the study failed. But it doesn't really. Of course it matters, but it doesn't matter to my point, which is that biomarkers have completely changed the way we do clinical trials. They've changed the way we do clinical trials because if we're using an amyloid-lowering drug, we want to make sure people have amyloid, and we want to make sure that the amyloid is actually being lowered. So, where does this leave us? Well, I think, especially if the aducanumab study is really positive, there are going to be more amyloid-lowering therapies. And now we can test people we can study people who are asymptomatic, who have these genetic risks, and we can study asymptomatic people who have positive amyloid biomarkers. So we don't even need to study individuals with symptoms. We can study people based entirely on their amyloid biomarkers. And then there are other potential therapies. So there's therapies that could lower tau, and we can measure the effects of these drugs on tau. We can look at lifestyle interventions. There's a large, you've heard about some of them. There's a large one that we're part of called the Pointer Study that is a multi-domain lifestyle intervention, and it'll be interesting to see if that works and whether it affects any of these biomarkers. And so biomarkers are going to be very important in selecting people and in monitoring treatment. So that's really it. I think I left a couple of minutes. I'll thank, this is my lab, terrific group of young people who make me look good. Thank you.